All right, class. Today we're getting ready to finish chapter 12. We start to make a transition and look at a few physics components. And we really start to look at Ohm's law. Well, Ohm's law really deals with electrical energy and voltage, current, and resistance. And so what is electrical energy? Well, electrical energy is the movement of charged particles. So the neuron activity is dependent upon the electrical current. And so we look at voltage first. And voltage is basically what we look at when we talk about potential energy. It's the amount of difference in the electrical charge between two places, and it's measured in volts or millivolts. And when we measure this, we're usually looking at the inside of the cell versus the outside of the cell. Now, the current is the actual movement of charged particles across the barrier that separates them. And this current can actually be harnessed to do work. And we see this as we have the current moves and generates the action potential. The action potential is actually performing the work of the body. And then we have the resistance. And the resistance is the opposition to the movement of those charged particles. And that resistance, in our case, is going to be a barrier or the plasma membrane. An increase in resistance, however, lowers the current. So when we look at Ohm's law is where the current equals the voltage divided by resistance. And when we compare or when we look at current, we're going to notice that it increases with a larger voltage and a smaller resistance. Now, as it applies to neurons, we look at charged particles are the ions, so our sodium and potassium ions. The current is then generated when these ions diffuse through our particular channels. The voltage then exists across the membrane due to the unequal distribution of the ions. Again, we've talked about how there is more sodium ions outside the cell and more potassium ions inside the cell. So this discrepancy and unequal distribution of the ions allows us to generate this voltage. The membrane itself then offers the resistance to ion flow. And this resistance changes due to the actions of our voltage-gated channels. And then the resistance decreases when the channels open, because at this point, we're now allowing the ions to move through those channels. And so there is no longer the same amount of resistance in the membrane. So when we look at some of the neurons at rest, we look at a couple different characteristics. One, we know that ions are unevenly distributed across the plasma membrane, and this is mainly due to the action of the pumps, particularly for our purposes, the sodium and potassium pump helps to maintain that uneven distribution. We're going to see that we have a higher concentration of potassium in the cytosol or within the cell in comparison to outside the cell or within that interstitial fluid. We also then see higher concentrations of sodium outside the cell, but we also see higher concentrations of chlorine and calcium in the interstitial fluid or outside the cell than what we do within the cytosol or inside the cell. Gated channels are then closed in the functional segments of the cell when neurons are at rest. And there is an electrical charge difference that's actually generated because of this unequal distribution and because of the movement of those ions through leakage channels across the membrane. The cytosol is relatively negative compared to the outside of the cell or that interstitial fluid. And the resting membrane potential, that RMP, is typically right around minus 70 millivolts. And again, we measure this through microelectrodes where we put one inside the cell and one outside the cell. And this is basically the utilization of a voltmeter. So this is a graphic of our neuron at rest. This gives us a really good idea of where a lot of these channels are, from our chemically gated chlorine channels to our voltage gated potassium channels, our voltage gated sodium channels, so on and so forth. It also shows us the receptive region, which is going to be the dendrites in the cell body. We have that initial segment, which is that axon hillock, that Y-shaped or V-shaped structure. We have our conductive segment, which is essentially the axon. And then we have the transmissive segment at the very end with the axon terminal and the synaptic bulb, where we're actually releasing the neurotransmitters from the cell. So again, this will be a good slide to either hit pause and just kind of review and look at and get a good idea of where a lot of these structures and channels are. So as we continue with our neurons at rest, we start to look a little bit more at that resting membrane potential. We have to understand that sodium and potassium are going to diffuse into and out of the cell based on their concentration gradients. But we also have to understand then the process of how sodium and potassium move based on the opening of our voltage and ligand gated channels. So potassium diffusion is the most important factor in setting the resting membrane potential, because what happens is potassium diffuses out of the cell due to its concentration gradient. Potassium diffusion out is limited, however, by the electrical gradient, which is the pull of the negative RMP on the positive ion. So if we look at if potassium were the only ion that was leaked, the RMP would be where the equilibrium of potassium concentration and electrical gradients are at equilibrium. And that would be about minus 90 millivolts. So again, if we kind of sum this up, we have potassium that diffuses out due to the concentration gradient of a higher concentration of potassium inside the cell versus outside the cell. So we're moving down the concentration gradient. However, the negative inside of the cell actually starts to pull some of those positive ions back in. And so we get this resting membrane where that pulling or that equilibrium point is reached. Since there are only a few sodium leakage channels, sodium also influences the RMP, although it's not as significant as what potassium is. Sodium diffuses into the cell due to its concentration gradient, as well as the electrical gradient. So remember, if the cell is more negative inside and sodium is outside the cell, which it is, and is a positive ion, you have the diffusion down its concentration gradient from high concentration outside the cell to low concentration inside the cell. But you also have the electrical gradient of a negative charge inside the cell, allowing that sodium to be pulled in. Again, positive and negative ions or positive and negative charges attract each other. So a negative charge also then pulls in a few extra sodium ions. The small leakage of sodium actually means that the resting membrane potential is a little bit more or a little less negative as opposed to where it would be at that minus 90. So instead of being minus 90 where potassium would be the only ion that's leaked, because we do have some sodium, that actually means that the RMP is actually a little less negative than what that minus 90 millivolts would be. So the resting membrane potential is minus 70. As we continue to look at the neurons at rest, we really start to look at the sodium-potassium pumps then as well and what their function is. By pushing 3 positive charges out and pushing in only 2, the pump contributes about minus 3 millivolts of the minus 70 total. So more importantly, it really helps to maintain the concentration gradients for these ions, as you notice that that 3 millivolts is pretty negligible in the entire minus 70. So the major purpose of the sodium potassium pump is really to help maintain the concentration gradients as opposed to the electrical gradients. So here we just take our voltmeter and we're taking a reading of a neuron at rest. You do get a chance to see all the channels that really are involved, from the sodium potassium pumps through the leakage channels for both sodium and for potassium. So here we get to take a look at the overview of the parts of the neuron, of which we've already looked at and seen a graphic before. Previous graphics may have been a little bit more detailed with the channels that were involved. However, in this case, we're really isolating these segments that are involved. And we'll end up talking about a little bit of what each of these is responsible for as we progress here in chapter 12. So when we look at the receptive segment, this is going to be the dendrites and the cell body component. We look at what graded potentials are. Now, graded potentials and action potentials are related. However, graded and action potentials are different. Graded potentials are going to be small, short-lived changes in the RMP with the following characteristics. They're established in the receptive segment by the opening of chemically gated ion channels. The local current changes are short-lived. They vary in degree of change and direction of change of the RMP. And what that means is they can be large or small in size, so they can have a large change in voltage or a small change in voltage. And they can cause a depolarization or a hyperpolarization. Now, a depolarization is where the cell is becoming more positive. A hyperpolarization is where the cell is becoming more negative. So with a depolarization, we're moving from a minus 70 RMP, and we're coming closer to threshold. So we can move to minus 65 millivolts or minus 60 or minus 55 millivolts as we come closer to that threshold stimulus. A hyperpolarization moves us farther away from the threshold. So at minus 70, we're at the RMP. With a hyperpolarization, we're moving to minus 75 or minus 80. So that is the difference between depolarization and hyperpolarization. Now, graded potentials are going to occur in the postsynaptic neuron, and these are called postsynaptic potentials. A postsynaptic potential that results in a depolarization is called an excitatory postsynaptic potential, or an EPSP. A postsynaptic potential resulting in a hyperpolarization is called an inhibitory postsynaptic potential, or an IPSP. So with an EPSP, we're actually becoming more positive. Again, becoming closer or coming closer to threshold. And this is what's going to be more excitatory. It has the greater potential to get us to an action potential. Whereas an inhibitory postsynaptic potential, or an IPSP, because it's a hyperpolarization, it's actually making us more negative, and it's taking us away from the threshold level. As this occurs, this is actually making it more difficult and more unlikely that we generate an action potential. Now, the one thing that occurs is that numerous postsynaptic potentials can be generated because the postsynaptic neuron can bind many neurotransmitter molecules simultaneously. It's all about the connection on the particular postsynaptic neuron and what channels are being stimulated at that particular location. So if we look at the generation of an EPSP, EPSPs are depolarizations. And so because they're depolarizations and they're becoming more, or the cells are becoming more positive, these are caused by an influx of sodium entry. So sodium is basically flooding into the particular cell. And so you can see it here, the generation of an EPSP. We have numerous, in this case, numerous neurons that are synapsing with the particular postsynaptic neuron. And as the neurotransmitter is released, it's causing the opening of these chemically gated cation channels. And these ion channels are actually allowing sodium to flood into the cell, which makes the cell more positive. So you can see the steps here. The sodium flows into the neuron, which is faster than the potassium flows out. The inside of the neuron becomes more positive or less negative, and this is a depolarization. And then the EPSP moves towards the initial segment. Because again, if we generate a signal large enough, we're actually able to generate an action potential at that axon hillock. So here we have our stimulus. Here's the distance of an EPSP. Again, you'll see that it's just a small depolarization. In this case, it doesn't reach threshold. And because of that, it diminishes over a distance and returns back to the resting membrane potential. So this was just one particular impulse, or one particular stimulus, I should say, at one particular location. If we look at the generation of an IPSP, these are the opposite. These are hyperpolarizations that are caused by potassium exit leaving the cell, becoming more negative, or chlorine entry, which you'll notice up here, the chlorine ion is a negative ion. So a negative ion coming into the cell also makes it more negative. So here you see the same steps, just with different ions. In this case, we have the neurotransmitter released. We bind to chemically gated channels. We have an efflux of sodium out, which makes the cell here more negative, or we have an influx of chlorine, which also makes it more negative. So the inside of the neuron becomes more negative, called an IPSP. You'll see a 2 millivolt change in this case. And then it does the same. The IPSP moves toward the initial segment. Here you'll see it in graphical representation. We have our stimulus. Here you'll see the hyperpolarization, or the cell becoming more negative. And then over distance, it diminishes and returns back to its resting membrane potential. On this screen, you'll see basically a connection. So here we have a single neuron. In this case, this is our postsynaptic neuron. But you'll notice that there are green connections and red connections, and you'll see this all over the place. So this particular neuron is being stimulated by a multitude of different neurons throughout the entire cell. And these are different connections that are created or formed in the body. Now you'll see some of them are going to be EPSPs. So some of them are going to be excitatory and stimulatory. Others here in red are going to be inhibitory. So as all of these EPSPs and IPSPs come in, again, on the red, they're IPSPs. On the green, they're EPSPs. As they all come in, they get smaller over distance. But when you have a bunch of them coming in all at the same time over a widespread area, or in rapid fire order from the same location, they can actually add up in what we call summate all the way down into this axon hillock. If the net change is enough to reach the threshold, we are able to then generate an action potential at this initiating segment of the axon hillock. So this is where we start talking about summation. And so the summation of EPSPs and IPSPs occur at the axon hillock. We're going to see the voltage changes, and it does this from the dendrites and the soma. And as these changes occur, they're added together, so they summate. Some of them may be more positive changes, some are more negative changes. But the sum of these changes may or may not reach the threshold membrane potential for initiating an action potential. The threshold is the minimum voltage change that's required to generate an action potential at that initial segment of the axon hillock. Typically, the threshold is about minus 55 millivolts. So we need approximately a 15 millivolt change in order for us to generate that particular action potential. And generally speaking then, multiple EPSPs must be added to reach threshold, because multiple IPSPs are going to take you farther and farther away from the threshold, and it basically makes it impossible to generate the action potential. If the threshold then is reached at the axon hillock, we stimulate the opening of our voltage-gated channels, particularly our voltage-gated sodium channels. And when we open these sodium channels, we get this flood of sodium into the cell, which now makes the cell more positive, and an action potential is generated. So we have a couple different types of summation that occur across space, but also across time. So with spatial summation, this is where we have multiple locations on a cell's receptive region receive the neurotransmitters simultaneously, and they generate postsynaptic potentials. So these are occurring over a wide area or over a space. Whereas temporal summation is slightly different. It's where we have a single presynaptic neuron repeatedly releases the neurotransmitter and actually produces multiple EPSPs within a very short period of time. So spatial summation is over a wide area or over space. Temporal summation is basically rapid fire order, almost like a machine gun at the exact same location. So here we have spatial summation. And again, we've taken that same graphic of multiple locations. You basically can essentially pick multiple green locations, if you will. So if we follow this table here, we have two simultaneous EPSPs summed together. So here we have P1, okay? Here we have P2. So we have one signal coming in at 1 over here. Here we have another signal coming in at number 2. As they add together, they actually add the voltage. So this is the size of P1. This is the size at P2. And this is what happens when we add them together. You'll notice that there's about a 2 or 3 millivolt change, 2 or 3 millivolt change. You add them together, you get approximately a 5 millivolt change. And so this is bringing us closer to threshold. Again, these are EPSPs. Now here we have the two simultaneous IPSPs summed together. Here we have postsynaptic potential at number 6. So we got one coming in there. And then also at number 7. So all the way over here. So as one comes into the cell body, another comes into the cell body. Again, the same thing. You'll notice in this case, we got about a 2 or 3 millivolt change, a 2 or 3 millivolt change towards the negative side. When you add them together, it produces a greater IPSP of about 5 millivolt difference. So now if we look at a simultaneous IPSP and EPSP, they're actually able to cancel each other out. Again, P1 and P6, they come in at the same time. One is excitatory, one's inhibitory. When you add them together, you basically get no change at the axon hillock. So this first row here, this is actually going to lead us closer to generating an action potential. The second row here is going to take us farther away from generating an action potential and is less likely to. In this case, we have it canceling each other out, so there's really a net of no change. Now, with temporal summation, these generate the signal at the same location. So these are postsynaptic potential 1. You'll see these are EPSPs. So we have one signal coming in, but then we have another one following it right away. You can have two separate ones that are further spaced apart. You get one signal, it comes back to resting. You get another signal. But what happens if they come closer together in more rapid fire succession? Here you get one, it starts to repolarize, but then another one. And so again, you see the summation of these together to bring us a little bit closer to threshold. This is what we call temporal summation. And you'll see the same thing happen with IPSPs. You're bringing it farther away from the threshold, which means we're farther away from generating an action potential. So now that was our receptive segment. What happens at the initial segment? Well, the initial segment is the area around the axon hillock. And we've talked about having to reach the threshold stimulus of that minus 55 millivolts. If we are able to do that, if we reach the threshold stimulus, an action potential is generated and is propagated down the axon without any loss in intensity. So one of the characteristics of an action potential is what you see up here. It's all or none. Okay, there is no in between. If you reach threshold, that action potential is going to spike all the way to positive 30. And it's going to do that all the way down the axon itself without losing any intensity. However, if the threshold is not reached, which means the stimulus basically is a sub-threshold stimulus, the voltage-gated channels are going to stay closed at the axon hillock and no action potential is generated. The axon, when we look at it, shows the same intensity of response to values greater than the threshold. Okay, So if our threshold is minus 55 and we have a signal that gets us to minus 55, and then on another axon we have a signal that gets us to minus 40, those differences don't matter, because essentially what we see is once we reach minus 55, or once we reach that threshold, we spike all the way to positive 30. So we're going to see the same action potential regardless of what that threshold stimulus actually is, so long as it reaches the threshold number. So again, it's very similar in this case to what occurs with a gun as far as the action potential. If you have a gun and you pull the trigger, you get sufficient pressure on the trigger, the gun is fired. If you pull the trigger and there's not enough pressure on it, there's no firing of the gun. There is no partial firing. So the firing of the gun is going to occur if the trigger is pulled hard enough, but it also doesn't matter if you reach the point of enough pressure, whether you squeeze it really soft and reach the threshold, or you squeeze it really hard and reach the threshold, the firing is still the same. Gun still fires, bullet still comes out. So it doesn't matter whether you pull it relatively slow to get to that point, or you pull it really quick and hard to get to that point. So now we move into the conductive segment, and the conductive segment is the axon. And this really includes the axolemma, because it's what conducts the action potential. The action potential involves a depolarization and a repolarization. Now, depolarization is a gain of positive charge as the sodium enters through the voltage-gated sodium channels. You'll notice that this is really similar essentially in how we've done all of our contraction components. If you look back at muscle contraction, this is basically the same as what we had with the sarcolemma and on the plasma membrane of the muscle cell. If the action potential is generated, it opens up voltage-gated sodium channels, sodium floods into the cell. It's the same thing here. Sodium floods into the cell, making it more positive. We then have the repolarization phase. And repolarization is the return to the negative potential as potassium exits through the voltage-gated potassium channels. So at this point, our voltage-gated sodium channels have closed up, and our voltage-gated potassium channels have begun to open. And now potassium exits the cell, and as we have positive ions leaving the cell, that's going to help the cell become more negative. The action potential is then propagated down the axon all the way down towards the synaptic knob. And once we get to the synaptic knob, we obviously are transitioning to a different segment of the neuron. But these voltage-gated channels are going to open sequentially down the axolemma so that the next section is able to be stimulated. And the propagation of this action potential or a nerve impulse is called a nerve signal. Okay? So a nerve signal and nerve impulse are really the same. And we can also call this essentially the propagation of the action potential. All three of those really are synonymous with each other. The propagation of an action potential, a nerve impulse, and a nerve signal all represent meaning the same thing. So how do we actually generate the action potential during depolarization? Well, there are a few steps in depolarization. So at the RMP, the voltage-gated channels are closed. However, once we reach that threshold number and sodium is allowed to enter from the adjacent regions, we're stimulating the voltage-gated sodium channels to open. So as more sodium floods in earlier, it's going to stimulate the opening of more voltage-gated channels a little bit further down the chain. And so this is kind of what we see with the action potential as it moves down the axon. Again, here's our voltage-gated sodium channels that are closed. And then down here, you start to see where they're actually opening. And this allows an increase in sodium into the cell at different points. So as sodium enters the axon, then it causes the membrane to have a more positive potential. And you can see that this is occurring as we reach threshold, and we're becoming more positive as we reach that positive 30. In step 4, then, the sodium channels close, becoming more inactive. And these are temporarily closed for this particular purpose. So as they close, then, you start to see that there's a decrease in the amount of sodium that's able to flood in. And so now we start to begin the next phase, which is going to be repolarization. So what then happens is that we repeat these first 4 steps in adjacent regions, and that actually allows, as you look down here in the bottom here, number 4 at my pointer, it allows the next phase to open, and the next phase, the next phase, and it allows that depolarization wave to continue all the way down the axon on the axolemma, all the way down to the synaptic knob. So then the next few steps then relate to repolarization. So with repolarization, we now have at the top of the peak, we now have our voltage-gated sodium channels that have started to close, but now we notice that we have our voltage-gated potassium channels begin to open. And so as those channels open, potassium then diffuses out of the cell, which now is making the cell a little bit more negative. And that's bringing us back down closer to our resting membrane potential. Now we'll find that the potassium channels stay open for a little bit longer past our resting membrane potential. And that allows an excess efflux of potassium out of the cell. And that actually makes the cell a little bit more negative than the resting membrane potential. And this is what we call hyperpolarization. So again, voltage-gated sodium channels close, voltage-gated potassium channels open, allowing an efflux of potassium out, makes the cell more negative. We have an excess efflux of potassium out of the cell, making the cell a little bit more negative than resting membrane potential, and that is called hyperpolarization. Eventually, the voltage-gated potassium channels close, and the RMP is reestablished and helped to be maintained by the continuous flow through leakage channels, but also through the sodium-potassium pump as well. And so then steps 5 to 7 repeat in the adjacent regions as the impulse moves towards the synaptic knob as well. And so you have this lead phase here of depolarization as it moves down the axon. However, right behind it is occurring repolarization. And so we have this wave that basically moves straight down the axon. So here are the events of an action potential. Again, you get a chance to look at all 6 phases or all 6 portions of this. We have it divided up into sections on the graph here. Here's our resting membrane potential of minus 70. You'll see that here we have these graded potentials. Again, whether we summate through spatial summation or we summate through temporal summation, understand that in order to get to threshold, we have to have a greater summation of EPSPs, which are excitatory and are bringing us closer to this threshold stimulus. So here we have our graded potentials in step 2. You'll notice that once we reach threshold, there is no middle ground. This is all or none. So if we stopped right here, it would come back down to resting. But since we did reach threshold, we go all the way up. This is depolarization in step 3 here. We reach the peak. We now get repolarization. So here was the opening of our voltage-gated sodium channels. Here's the opening of our voltage-gated potassium channels as we go through repolarization, all the way down to our resting membrane point. We have that increased efflux of potassium out of the cell, and this causes our cell to become more negative. And this is that hyperpolarization phase before we reach back to our resting membrane potential due to the sodium-potassium pumps and some of our leakage channels. As we continue with our conductive segment, we really have to look at the refractory period. Now, there are 2 different types of refractory periods. But what is the refractory period? Well, it's the period of time after the start of the action potential when it's either impossible or difficult to fire another action potential. The absolute refractory period is about 1 millisecond, and it's at this point where no additional stimulus can initiate another action potential. It's in this phase where our sodium channels are open and then inactivated. So this is basically through the phase of depolarization and then partway into repolarization. And the absolute refractory period purpose is to ensure that the propagation of the action potential goes toward the synaptic knob and doesn't actually reverse in its direction. Now, the relative refractory period is just after the absolute period. It's at this point now where another action potential is possible because the sodium channel, the voltage gated sodium channels have reset. However, the minimum stimulus strength is actually a little bit greater than what it was when we're at the resting membrane point. It's at this point where some potassium channels are still open, the cell is still slightly hyperpolarized, and is further away from the threshold stimulus. So here you see the absolute refractory period. Again, you'll notice that it includes depolarization once you reach the threshold mark, and it goes into a big chunk of repolarization. The refractory or the relative refractory period is basically towards the end then, once we reach the threshold of repolarization, and it includes the hyperpolarization phase. So remember, absolute refractory period, there is no possible way to generate another action potential. The relative refractory period, we can, it just requires a significantly stronger stimulus for us to be able to do that. So as we continue with the conductive segment, we also look at the conduction process and how the axon actually is able to conduct the signal down the axon. And so we look at continuous versus saltatory conduction. So with continuous conduction, it's going to occur on the unmyelinated axons. And so what happens is the charge opens the voltage gated channels, it allows the charges to enter, and it spreads to the adjacent regions and opens more channels sequentially, similar to what you would expect, okay, with the process of propagating an action potential. However, saltatory conduction occurs on the myelinated axons, and it's slightly different. If you remember back to myelinated axons, and we have the Schwann cells that make up these kind of little bubbles, and they have these spaces called our nodes of Ranvier, or our neurofibril nodes. So what happens is the action potential occurs only at these particular nodes. And it's at these nodes where our voltage gated channels are actually present. Okay? And so the voltage gated channels are actually concentrated at these nodes of Ranvier. As sodium enters at a node, it actually starts a rapid positive current down the inside of the axon's myelinated region. And so it moves the signal and moves the current inside the area that's surrounded by our Schwann cell, or around the area that has the myelination. As it goes across that myelinated area, it actually becomes just a little bit weaker, but it's still strong enough to open the voltage gated channels at the next node. The full action potential occurs then at the node, and the process repeats. And so what it actually appears like then is that the signal is really jumping from node to node. And this saltatory conduction then is much faster than continuous conduction, and the myelinated cells actually use less ATP to maintain the resting membrane potential. And so as it moves from one node to the next, it actually is able to transition itself down the axon at a higher rate of speed. So here we have continuous conduction. Again, we don't have the myelination, so we're literally moving from the next patch of the cell, and we're changing the voltage in that particular area. With saltatory conduction, it's a little bit different. You'll notice that we are going to have channels literally every little step of the way on an unmyelinated axon. But here we have the channels only in these nodes. And so you'll see the voltage gated sodium channels open. It allows the action potential to be generated at the node. And then it zings its way all the way through the myelinated section until it stimulates the next node. And then it zings its way all the way through the myelin sheath and stimulates the next node. And this is how it's actually able to move down the axon at a higher rate of speed, because the Schwann cells in the myelination help to insulate it, and so the cell doesn't actually lose a lot of the excess neurons, or excuse me, a lot of the excess ions around that particular area. So that then moves us to the transmissive segment. And so now we're at the synaptic knob, where we're going to go ahead and release our neurotransmitter. And again, this will be very similar to what we've already talked about in our muscle contraction. We have the arrival of the action potential, and it's going to open voltage-gated calcium channels. The calcium channels allow the diffusion into the cell of calcium. Now, calcium is going to bind to the proteins that are associated with the synaptic vesicles, and it's going to trigger the process of exocytosis. So now these vesicles are actually going to move towards the end of the synaptic knob. They're going to fuse with the membrane, and then the neurotransmitter is released into the cleft. Now remember, this neurotransmitter can be excitatory or inhibitory. In muscle cells, it's excitatory. So we may release a neurotransmitter that actually causes some sort of response on the postsynaptic neuron that generates a new signal, or it can actually inhibit the production of a signal. So subsequently, the neurotransmitter then binds to the postsynaptic receptors on those ligand-gated channels. And again, we find out that neurons can actually synthesize more than one neurotransmitter. However, only one type is going to be released at a time. So it's going to depend on whether the neuron is trying to generate an excitatory response or an inhibitory response. And whichever neurotransmitter is stimulated or released is dependent on the frequency of the action potentials that reach the synaptic knob. So how fast they're actually coming in tells us what neurotransmitter we need to release. So here we have our chemical synapse utilizing the chemical or the neurotransmitter. Again, here we have our nerve signal or the action potential that comes down and stimulates voltage-gated calcium channels. It allows calcium to then flood into the cell. It binds to the proteins on the vesicles, which cause the vesicles to move toward the surface of the synaptic bulb. And it causes the release of the neurotransmitter by the process of exocytosis. Our neurotransmitter then crosses the synaptic cleft, and it binds to our ligand-gated channels that open up and cause some sort of ion to allow to flood into the cell. So let's compare graded potentials versus action potentials. And this will be a really good slide for us to really understand the difference. So if you've had a little confusion up to now as to what they are, hopefully this slide really helps to clear a lot of that up. So a graded potential occurs in the neuron's receptive region due to the ion flow through the chemically gated channels. So these are occurring really in our dendrites and our cell bodies of the neuron. Again, they can be positive or negative changes in the charge, so that we have EPSPs and IPSPs. They're called graded potentials because they are graded. They can have larger potential change to stronger stimuli, or they can be relatively small. Again, they're always going to typically fall under the distance needed to generate the action potential or the threshold. But what happens is they are able to add together. And then lastly, they're very local. So they only travel short distances, and they get smaller or they diffuse over that distance. So they may come in on the dendrite, and as it travels from the dendrite in toward the cell body, it starts to degrade and get smaller. And by the time it actually reaches a part of the cell body, it may not actually be there anymore. And so again, this is how they only travel short distances. Now, action potentials, on the other hand, occur on the neuron's conductive region, or the axon. And it does this due to ion flow through the voltage-gated channels, particularly with our sodium initially. So it involves the depolarization phase, which is sodium flooding into the cell. And then it involves our repolarization phase, which is potassium out of the cell. Again, action potentials are all or none once the threshold is reached. So they don't change in size. It goes from the threshold level of minus 55 all the way up to positive 30 every single time. And that's the difference between an action potential and a graded potential, or one of the differences. It also propagates the entire axon and all the way down to the synaptic knob, where again, a graded potential is short distance. It may come in on a dendrite and be gone by the time it hits the cell body. When we look at conduction speeds, we have to look at the axon as well as the myelination involved. So everything up to this point was the generation of the action potential, but now we look at how fast the action potential is able to move. And so when we look at the thicker fibers, they're actually going to be able to conduct the signal or the action potential faster than our thin ones, mainly because the thick axons offer less resistance to current flow all the way down the axon. Myelinated fibers also conduct the impulse much faster than unmyelinated ones, mainly because the current flow under the myelin is very fast. So the signal really is occurring between, or I should say, the action potential is actually occurring at the nodes, and it allows the flow of the current to occur in the myelinated sections between the nodes. So within the velocity of the action potential, not only do we look at nerve diameter and myelination, but we also look at the different types of fiber groups. So a nerve fiber is the axon and its myelin sheath. So it's really the conductive region. We have a couple different types of nerve fibers. We have a group A type fiber. This is the conduction velocity that's really fast, and it moves at about 150 meters per second. These tend to be large diameter myelin fibers. And most of our somatic sensory neurons are group A fibers, as well as all of our somatic motor neurons are group A fibers. Again, remember what you should think of when you see somatic. You should be thinking skin, skeletal muscle, and joint. So these are sensory neurons coming from skin, skeletal muscle, and joint. And then we also have motor neurons basically going to our skin, skeletal muscle, and joint areas. So that's basically innervating our skeletal muscle. Now, group B fibers are still relatively fast, but they're going to conduct the signal at about 15 meters per second. Group C fibers are the slowest. These move the signal at about 1 meter per second. So these are small diameter and are unmyelinated cells. They can be myelinated, but it's really kind of an and or. Some are going to be myelinated, some are unmyelinated. And these are going to include a lot of our visceral neurons, as well as some of our somatic sensory neurons from the skin. Okay, so there will be some group B and C fibers in this category as well. So let's look at the frequency of the action potential, because the frequency of the action potential does tell us a lot in what type of signal we're trying to generate. When we look at the impulse frequency, this is where we have a certain number of action potentials per second. And impulse frequency can vary. Although the impulse amplitude or the action potential or the change in voltage is constant, we have the same height, so it goes from that threshold to positive 30. that particular part of the action potential is constant. However, the firing frequency, or how many action potentials we have in a period of time, is going to vary with stimulus strength. So when we look at an example, bright lights are actually able to cause a faster firing frequency on the optic nerve than dim lights. And so when the motor nerves fire at faster frequency, it actually causes the muscles to generate more tension. So light tension means slow frequency. More tension or stronger contraction means a greater frequency. And so for neurons that can use different neurotransmitters, the firing frequency then can influence the type of the neurotransmitter that gets released. So let's look at these neurotransmitters. So neurotransmitters are synthesized by the neurons, and they're stored within the vesicles in a synaptic knob. In the muscle cell, we talked a lot about acetylcholine. That is one example of a neurotransmitter. Norepinephrine, which we've kind of touched on periodically, is also an example of a neurotransmitter. And those are probably the two most common types that we will really discuss. Our neurotransmitters are released from the vesicles when the action potential triggers the calcium entry into the synaptic knob, and it binds to a receptor on the target cell. This binding actually then triggers a physiologic response of the target cells. Now, as far as the neurotransmitters go, we've identified approximately 100 different neurotransmitters, and we've also then classified them into different groups. So there are four main chemical classes of neurotransmitters. Acetylcholine is one of them, and the structure differs substantially from other neurotransmitters. And obviously, this is the one that we talk about with the muscle contractions. We also have biogenic amines, and this is an amino acid that's slightly modified to synthesize the transmitter. Examples would be catecholamines, such as dopamine. These are made from tyrosine. And then we have indolamines, such as serotonin, which are made from histidine or tryptophan. Amino acids are another chemical class. These include common neurotransmitters called glutamate, glycine, and GABA. And then we also have our neuropeptides, and these are chains of amino acids, approximately 2 to 40 amino acids long, that include endorphins and substance P. Now, I'm not so concerned about all of these, making sure that you know them and memorize them. There are a few that are really important, though. We'll talk a little bit more about dopamine later, so I'm not too concerned about that at this moment. We do want to know acetylcholine because, again, that's involved in our muscle contraction. We also want to know endorphins and substance P, okay? So we're going to make sure that we do know these particular functions. So endorphins, as you probably already know, endorphins are the body's natural painkiller. They have a very similar function from a physiological component as what opioids do. And so it's kind of the body's natural opioid, if you will. It's the body's way of dimming or taking care of certain pain avenues. Whereas substance P, on the other hand, kind of does the opposite. Whereas endorphins are the body's natural painkiller, substance P actually stimulates the pain response, okay? So we have two separate neurotransmitters here that have basically opposite functions. And these are the two, at least on this particular page, that we're going to want to make sure that we understand. Endorphins and substance P. So make sure that if you haven't, you rewind, you at least jot down the functions. Endorphins, natural painkiller. Substance P, they kind of stimulate the pain pathway. Okay, they mediate pain signals. So we can also have different classifications for neurotransmitters based on their function and how they affect the membrane potential. So we can have excitatory neurotransmitters, and they're going to cause EPSPs. They're excitatory. Inhibitory transmitters cause IPSPs. Now, you'll notice that we have kind of this statement underneath it. Some neurotransmitters can excite some targets and inhibit other targets, depending on the specific receptor present on the target cell. So they can have multiple functions depending on the cell that they're trying to target. We also classify them by action. So neurotransmitters can have direct action. Direct transmitters bind to the receptors that are chemically gated channels, and they provide an immediate postsynaptic potential. An indirect transmitter binds to receptors, and they involve second messengers, including G proteins. However, one of the differences between the two is that direct neurotransmitters, they're going to typically bind to immediate response. They're going to have one particular response. With the indirect transmitters, because we're involving second messengers, they can actually lead to more diverse effects in their response. So here's just kind of a flow chart. It gives us the classification based on chemical structure and the classification based on function from their effects, whether they're excitatory and inhibitory, or their actions, whether they're direct action or indirect action. Again, we said we talk about acetylcholine a little bit more in depth, and it is the best characterized neurotransmitter. Again, we talk about it with muscle contraction. We also talk about it when we get into the CNS. So in the PNS, the peripheral nervous system, it's used to stimulate skeletal muscle. In the CNS, it's actually used to increase arousal and awareness. It's synthesized from acetate and choline and is stored in synaptic vesicles within that synaptic bulb. The action potential that arrives at the presynaptic neuron triggers its release into the synaptic cleft. Some of the acetylcholine then attaches briefly to the postsynaptic receptors. And if we look at the breakdown of it, again, we've talked about all of these. Acetylcholine is then cleared from the cleft by being broken down to acetate and choline by acetylcholine esterase. Acetate and choline then are taken back up by the presynaptic cell, and they're essentially recycled. Now, their effect on target cells depend on the receptor that's actually present. And these are going to include nicotinic receptors and muscarinic receptors. But again, this slide in particular really isn't anything new compared to what we've already talked about with skeletal muscle and the neuromuscular junction. All of these components really are, or all these characteristics are really the same characteristics that we talked about when we discussed the neuromuscular junction. So one of the things that we haven't really gone into great depth yet is how do we remove the neurotransmitters from the synaptic cleft? And it can occur in varied ways. Well, when we look at acetylcholine, we know that we have the enzyme acetylcholine esterase. So we know that one of the ways is that there's an enzyme that can come in and degrade the neurotransmitter. We can also have that neurotransmitter be taken back up into the presynaptic neuron. And this is called reuptake. And so not only do we have something that's breaking it down, we also can just take it right back into the synaptic bulb. And then the last way is some of the neurotransmitter just diffuses away from the synapse and is reabsorbed by glial cells. Remember, our synapse is a fluid-filled space. We're not having a direct connection when we look at these particular types of synapses. And so there is space for that neurotransmitter to escape from that area. Now, one of the things about neurotransmitters is that some drugs have their effects by influencing the transmitter removal. For example, we have selective serotonin reuptake inhibitors, and these are going to be things that help to treat depression. We also have things that help to treat Alzheimer's, for example, And these are going to include galantamine hydrobromide, and it's an acetylcholine esterase inhibitor that is used to treat Alzheimer's. And so if you think about what this term means here, when we talk about an acetylcholine esterase inhibitor, basically what we're doing is we are inhibiting acetylcholine esterase from coming in and breaking down acetylcholine, which actually allows us then to help maintain a little bit more of a stimulation of particular neurons. So here we look at acetylcholine release, removal from the synaptic cleft, and its action. So you'll see the nerve signal would come in, it would open voltage-gated calcium channels, calcium would flood in, causing the synaptic vesicles to move toward the end of the synaptic bulb, where it's released or releases its contents through exocytosis. In this case, the contents are acetylcholine. Acetylcholine crosses the synaptic cleft and can act in two different ways. It can act in a direct action or indirect action. By acting in the direct action, we have nicotinic receptors. Indirect action, we have muscarinic receptors that stimulate or activate a G protein and a second messenger. In this case, we have sodium that floods into the cell. It becomes more positive. In this case, it also allows sodium to potentially flood in, but we also have a process that's occurring that allows for an EPSP or allows for an IPSP, okay? So again, it kind of depends on what type of receptor is found on this particular target cell. So now that we've kind of discussed essentially the entire process of the action potential down a neuron, how it interacts with synapses, we now look at how groups of neurons start to work together. And these are called neuronal pools. And we see these really in the CNS. And so these neuronal pools are groups of neurons that are arranged in a specific pattern. There are four types of circuits: a converging circuit, a diverging circuit, a reverberating circuit, or a parallel after discharge circuit. Now these pools may be localized or they can be distributed in several regions of the CNS. So if we look at a converging circuit, you'll notice that here we have multiple neurons that all kind of converge into one neuron. So the input converges at a single postsynaptic neuron. So the example of this type of circuit would be where we have multiple sensory inputs that synapse on the neurons in the salivary nucleus. So we can have sights, sounds, and smells of cooking all lead to one output, and that can be salivation. So we can have sensory neurons for sight, looking at food, sensory neurons for sound, hearing the cooking of food, sensory neurons for smells coming in, and all those put together can all lead us to, even if we have them separately, can all lead to one response, and that would be of salivation. Our next type is a diverging circuit. Now this is kind of the opposite. Instead of converging into one location, we now go the opposite direction. We have one signal moving out that synapses with multiple neurons that then send more signals out and gets us more outputs. Okay, so the signal basically is getting wider. So a diverging circuit spreads information from one presynaptic neuron to several postsynaptic neurons. Our example would be where we have neurons in the brain that control walking, and they're going to send commands to several different muscles for proper posture, balance, and motion. Again, so we have one function or one action, which is walking, but we have to stimulate numerous muscles and limbs and areas of the body in order for us to succeed with walking. Our third type is a reverberating circuit. Now, a reverberating circuit uses feedback to produce repeated cyclical activity. And once it's started, it stays active until there is an inhibitory stimulus or synaptic fatigue. And so we look at the breathing cycle, and it allows us to keep breathing regularly during sleep. So you'll see that we have the Output, we have the pathway that basically comes back around and comes back down this way, and you see the cycle of it continuing. It reverberates around the pathway or around that circuit. We also then have our last one is a parallel after discharge circuit. In this circuit, the input is transmitted simultaneously along several paths to a postsynaptic cell. Since the paths vary in number of synapses, the signal arrives at the postsynaptic cell at various times. And it's at this particular type of circuit that's believed to be involved in higher order thinking. Okay? And you can see the input here, it goes along different pathways. It's kind of parallel after it discharges until we get to one particular output. Now let's use the parallel after discharge circuit as our attendance code words for the day. Okay? So parallel after discharge is going to be the code words for today's lecture.